Okay, let's dive right into this explainer and crack the case of whether we can actually, truly bring dinosaurs back from extinction. I mean, it's a topic that's absolutely captivated us for decades, right? Blurring the lines between sci-fi and biological reality. So today we're going to sift through the actual facts, the hard limitations, and honestly, some mind-bending new strategies scientists are proposing right now to piece together our prehistoric past. Let's get into it. So, is a real-life Jurassic Park actually possible? It's the ultimate pop culture question. For years, the idea of extracting dinosaur blood from a prehistoric mosquito trapped in amber, we've all seen the movie, that was the gold standard for how we might clone a T-Rex. But, as we're going to see, the biological reality of our planet makes this Hollywood dream virtually impossible. Yet, the actual science of what we can do and where we're currently looking for dinosaur genetics, it's somehow even more fascinating than the movies.
Now, the central friction of our entire detective story lies right here. In recent years, paleontologists achieved the literal unthinkable. They actually found preserved soft tissue, we're talking collagen and blood vessel fragments, inside perfectly preserved 68-million-year-old T-Rex bones. Absolute massive breakthrough. But here's the catch. Despite having these physical remnants, the genetic blueprint, the actual DNA, is completely missing.
To understand why that blueprint is missing, we have to look at this number, 521 years. This is the biological half-life of DNA. Even under the absolute most perfect, freezing, stable conditions imaginable, the chemical bonds in DNA break down incredibly fast. It completely shatters that mosquito and amber myth. You see, amber just isn't perfectly airtight, and DNA decays through oxidation no matter what.
And this timeline perfectly illustrates the stark reality. The oldest DNA science has ever successfully recovered is just over 1 million years old, and that came from a frozen Siberian mammoth. At the absolute theoretical maximum, every last bond of a DNA strand would turn to dust after 6.8 million years. Now, the non-avian dinosaurs, they went extinct 65 million years ago. So they missed the DNA expiration date by nearly 60 million years. There is simply no intact dinosaur DNA left on Earth, period.
Now, you might be wondering, how do we know exactly how impossibly old these fossils are? Well, we don't use carbon dating. Carbon-14 decays so incredibly quickly that it's useless for anything older than 50,000 years. Instead, scientists use radioactive dating of elements like uranium-238 and potassium-40, which have half-lives of millions or even billions of years. And since these elements don't exist in the sedimentary rock where fossils actually form, researchers date the igneous rock, like layers of volcanic ash, found directly above and below the dinosaur fossil. This bracketing acts like a geological sandwich, proving beyond a shadow of a doubt exactly when these creatures lived and why their DNA is long gone.
So if the ancient DNA is completely gone, how in the world do we reconstruct a dinosaur? Well, we have to look at their living descendants. You see, evolution acts less like a giant eraser and more like an overwritten manuscript. Old genetic instructions are rarely just deleted. They're simply layered over, silenced, or repurposed by new adaptations.
And the crucial point here, and this is genuinely mind-blowing, is that birds aren't just related to dinosaurs. They physically are dinosaurs. In fact, because the theropod evolutionary line that led to birds is so incredibly direct, the genome of a modern chicken is actually closer to a Tyrannosaurus rex than a T-Rex's genome was to a Stegosaurus. We literally share the planet with highly evolved feathered dinosaurs right now.
Because birds carry this layered genetic history, nature occasionally glitches, producing what we call an atavism. This is when an ancient, silenced trait suddenly reemerges. We've actually seen mutant chickens naturally develop teeth, or horses born with extra toes reflecting their ancient ancestors. And scientists are currently trying to actively control these atavisms. Through initiatives often dubbed the Dino Chicken Project, they're attempting to engineer a chicken with a long tail, a toothed snout, and clawed forelimbs, simply by reactivating dormant traits already sitting right there inside the bird's egg.
If you're a tech person, think of the modern chicken genome like Microsoft Word version 7. It runs perfectly fine as a modern program, but buried deep down within its programming architecture is the legacy code from version 3. The instructions for a dinosaur's tail or teeth are still hiding right there in the bird's DNA. They've just been turned off by millions of years of evolutionary software updates.
Building on this genetic legacy, scientists have bioinformatically reconstructed the broader Archosaurian template, the common diapsid ancestor, by comparing the genomes of living birds and crocodilians. And what they found is absolutely astonishing. They discovered an incredibly stable genome structure consisting of about 10 pairs of macrochromosomes and roughly 30 pairs of tiny microchromosomes.
What's really amazing about this diapsid karyotype, this specific chromosomal structure, is that it has been locked in place for roughly 255 million years. It formed before turtles and birds even diverged. And this proves that the tiny, compact microchromosomes we see in birds today didn't shrink just so birds could become lightweight enough to fly. This genomic structure was established millions of years before pterosaurs or birds ever took to the skies. It is a highly stable, ancient blueprint.
But here is the massive limitation. Reactivating chicken DNA or computing a generic archosaur template can really only ever give us a generalized, generic dinosaur. It can never give us the specific, unique genes that made a triceratops, or an ankylosaurus, an ankylosaurus. Those unique genetic branches are cut off. So how do we find those missing pieces?
Well, this brings us to the wildest, most cutting-edge theory from our source material, the genetic shrapnel strategy. It's a form of forensic paleogenomics. The idea is that we might actually be able to find real microscopic fragments of dinosaur DNA buried like shrapnel, not in other animals, but in the modern genomes of plants and fungi.
To understand how dinosaur DNA could possibly end up in a plant, you need to know about horizontal gene transfer, or HGT. Normally, DNA moves vertically, right? From parent to offspring. But HGT is like a biological backdoor. Viruses, bacteria, and parasites can occasionally act as vectors, physically carrying genetic material from one completely unrelated organism to another, essentially inserting jumping genes right across the boundaries of species.
So picture the warm, humid, teeming ecosystem of the Mesozoic era. Step 1. A tick or a microbe feeds on the blood of a dinosaur. Step 2. That microbe then interacts with a local plant or fungus, acting as a viral vector that accidentally splices a tiny fragment of dinosaur DNA into the plant cells. Step 3. That dinosaur DNA embeds into the plant's dark genome, the non-coding junk DNA, and the plant has been actively replicating and preserving that genetic shrapnel as its own for the last 65 million years.
And guys, this isn't just science fiction. Researchers know exactly where to look for this shrapnel today. They're targeting candidate hosts that survive the asteroid impact and have massive, slow-evolving genomes. Fungi are great candidates. Certain ancient ticks that are basically living fossils are another. But the absolute best hiding spots? Gymnosperms. Plants like pines, conifers, and ginkgos. They have enormous dark genomes that act essentially as giant tape recorders of ancient biological interactions.
Now, you might think the odds of a microbe perfectly transferring a useful piece of dinosaur DNA into a pine tree are impossibly low. And yeah, they are. But consider this number, 2.5 billion. That's the estimated number of adult T-Rexes that lived and breathed over their entire 2-million-year reign. The sheer, immense volume of biological activity across hundreds of millions of years means there were literally billions upon billions of chances for horizontal gene transfer to occur.
But let's say we pull it off. Let's say we actually daisy-chain the genetic shrapnel from a pine tree with the legacy code of a chicken, and we get a complete Triceratops genome. We still hit a massive physical wall. Modern cloning, like what we did with Dolly the sheep, relies on replacing a mammalian cell nucleus and using a living host mother. But birds and dinosaurs lay eggs. Their egg nuclei are tiny, microscopic specks lost in a giant sea of yolk, making them currently impossible to manually replace. Plus, dinosaurs incubated externally, meaning we have no surrogate mothers alive today that could safely bring a dinosaur embryo to term.
And what if we could magically bypass all those cloning hurdles? We still have to face the ecological reality. Earth has moved on. The climate, the plants, the atmosphere, and most importantly, the microscopic pathogens of the 21st century are completely alien to a creature from the Cretaceous period. Resurrecting a dinosaur wouldn't be a triumphant return. It would be like dropping an invasive species onto an alien planet without a spacesuit. They have zero immunities to our modern world.
And hey, the danger absolutely goes both ways. Remember that dark genome we talked about earlier? Well, animal DNA is absolutely littered with ancient dormant viruses. If we synthesize and boot up a dinosaur genome, what happens if we accidentally awaken a prehistoric virus that's been dormant for 65 million years? We could be introducing a pathogen that modern mammals, including us humans, have absolutely zero evolutionary defense against.
Which brings us to a really fascinating philosophical wall, perfectly summarized by Dr. Ian Malcolm. Your scientists were so preoccupied with whether or not they could, they didn't stop to think if they should. The drive to achieve this is an incredible testament to human curiosity and our desire to master genomics, but de-extinction on this massive scale forces a really tough conversation about what our actual goal is. Are we just creating an isolated zoo attraction to see if we can? It's a real conversation scientists are having right now as this technology inches closer to reality.
And that's really what you should take away from all this.
The biological detective work currently happening, sequencing ancient ticks and mining the genomes of conifers to find the literal ghosts of dinosaurs, is an absolute triumph of modern science. It teaches us profound things about evolution, but it leaves us with a lingering, kind of heavy question. Even if we perfectly map the past and assemble the ultimate genetic puzzle, do we really want to pull the trigger and force the past into our present? I'll let you be the judge of that. Thanks for joining me on this explainer. Keep asking questions, and I'll catch you next time.